इलेक्ट्रोफोरेसिस के बाद सदर्न ब्लॉटिंग का प्रोसेस को जब हम लेके चलना चाहते हैं तो उसमें सबसे पहला स्टेप ट्रांसफ़र ऑफ डीएनए फ्रॉम जेल टू द सॉलिड स्पोर्ट या मेम्ब्रेन के ऊपर आता है। इस पर्पस के लिए हमारे पास डिफरेंट मेथड्स अवेलेबल हैं जो कि लैब में यूज़ किए जाते हैं। दैट includes the upward capillary transfer, downward capillary transfer, then simultaneous transfer to the two membranes using an electric current for electrophoretic transfer and vacuum transfer. इस प्रेजेंटेशन में हम बात करेंगे upward capillary transfer की। Upward capillary transfer, as the name indicates, it involves a capillary action. And the flow of the liquid is from bottom towards top. If you look in this picture, this is a setup that is established for upward capillary transfer. In this setup, we have a reservoir of a buffer at the bottom, and to this reservoir there is dipped a filter paper, the Wattsman filter paper, which crosses a glass plate or the solid sport, so that the edges of this filter paper are dipped in the reservoir. अब जब इसके किनारे इस फिल्टर पेपर के रेजरवायर में डिप हैं तो अल्टीमेटली क्या होगा, एब्जॉर्बेंस कैपेसिटी की वजह से जो रेजरवायर का सॉल्ट सोल्यूशन है दैट इज गोइंग टू ट्रेवल थ्रू दिस Wattsman paper, and then above this Wattsman paper we have our gel through which or from where we want the DNA sample to be transferred to the membrane. Right above this gel, this green color shows the presence of the solid sport, that is nylon membrane. Then there are two pieces which are shown here in blue color. These two pieces are of again Wattsman paper which are soaked in diluted solution. The buffer SSC buffer. SSC is a salt buffer that have the sodium chloride and sodium citrate in it. And above these two paper we have a stack of paper towels. These are totally dry paper towels, and at the top we have a glass plate and then a source of weight that should be equally distributed. Now this setup is going to create a column, and the liquid or the buffer from the reservoir will start moving due to capillary action from bottom towards the top, because at the top we have some dry paper towels. This buffer has to pass through the gel, and when this ionic solution is passing through the gel, it along with it carries the DNA through the gel, and that DNA is shifted over the nylon membrane. When this DNA is shifted to the nylon membrane, then basically we are going to achieve our target, that transfer of the DNA from gel to the nylon membrane or solid sport. यहाँ पे एक चीज़ देखने वाली है, the wells are like opposite और upside down. अगर आपको याद हो, हमने बात की थी हॉरिजॉन्टल जेल इलेक्ट्रोफोरेसिस की, जिसमें सैंपल हम टॉप से लोड करते थे और सैंपल डीएनए का जो है वो वेल में बैठ जाता था. इट मीन्स द डीएनए इज मोर क्लोज टुवर्ड्स द बॉटम. सो इफ वी वांट टू टेक द डीएनए सैंपल आउट फ्रॉम द जेल, वी शुड पुट द जेल upside down so that the DNA should be near to this top, and then we can place our nylon membrane over it. The principle in this case is capillary action. As I just mentioned, the concentrated solution starts moving through the gel towards the tower, up side the tower, and taking along the DNA with it. Now the question comes, the rate of DNA transfer. How much time it will take to transfer the DNA from the gel to the membrane? It depends on the size of the fragment, and it depends on the agarose concentration. If we are working with the DNA fragments which are smaller in size, less than 1 kilobase, then in that case, and they are run at in a gel of 0.7% agarose gel, then the transfer time is almost 1 hour or less than 1 hour. But if the fragment of DNA are larger, then for example more than 15 kilobases, then it can take up to 18 hours to transfer, and even the transfer is not still complete. in such longer transfer, the efficiency is going to be affected. For the reason, when the buffer from the reservoir moves up through the gel, it not only takes along with the DNA, but DNA के अलावा जो जेल का लिक्विड पोर्शन है उसको भी साथ में शिफ्ट कर दिया जाता है. अल्टीमेटली जो आपकी सेमी सॉलिड जेल होती है अगर उसकी, उसमें कुछ रबर टाइप का एक टेक्सचर आ जाता है. When the gel is going to get a rubber type texture, then it is very difficult that the liquid can further pass through and can perform its function. So in that case, we have to find out some alternatives. Either break down the longer segments into the smaller segment, or we have to proceed further with little amounts of DNA transferred on the membrane. So the solution of this problem is that when we are doing the electrophoresis, the DNA which is found in the gel, it can be treated with an acid and a base. That is going to cause acid-base hydrolysis. Treatment of that DNA which is in the gel with a weak acid is going to cause depurination. From random places, the purines are going to be removed. Ultimately, what is going to happen that when we are going to treat it with a strong base, from all those points where the depurination has been done, the DNA is going to break down. Like if you look at this drawing, treatment of DNA by weak acid is going to depurinate at this point. Then treatment with strong bases is going to cleave this DNA into 3 segments. And in this case, we can proceed further, and these small fragments can easily be transferred to the solid sport. The fragments which are around 1 kilobase in length, they are efficiently transferred from the gel to the solid sport, and their efficiency is much better as compared to the long sized DNA molecules.